This is a presentation for BIOCHEM424 by Alon, Yoonjae, Stephanie, and Evan. We're presenting the research question, is genetic silencing of BCL11A a viable method of treating sickle cell anemia? In its native structure, hemoglobin has 4 subunits consisting of 2 alpha and 2 beta subunits with 4 heme groups that can bind iron so that oxygen can transport throughout the body. In sickle hemoglobin, or HbS hemoglobin, there's a mutation that leads to the substitution of glutamic acid to valine in one of the beta subunits, and this leads to a hydrophobic residue being on the outside of the protein, which causes hemoglobin to have a sickle shape, and this can cause a lot of different symptoms in the body because hemoglobin will now form long chains that lead to blood clotting and can cause painful, swollen hands and feet, jaundice, fatigue, as well as irritability.
The treatment proposed in the paper was silencing BCL11A gene. There is a term that used throughout the paper called HbF. HbF is a beta hemoglobin that binds oxygen molecules and transports it through the bloodstream. In infants with sickle cell disease, it is shown to alleviate sickle cell symptoms, whereas in older kids or adults, higher HbF level leads to fewer symptoms. Sankara et al. 2022 paper indicated that about 20% HbF levels within all blood leads to alleviation of disease symptoms. BCL11A gene is a potent silencer of HbF in mouse models, which controls the beta-globin gene cluster in concert with other factors. The overall goal of the experiment was to raise the HbF level so that HbS proportion decreases, and in order to do so, they proposed HbF induction with BCL11A gene silencing.
The researchers used a lentivirus encoding a short hairpin microRNA that was specific to the BCL11A promoter region. The specific shRNA was called D12G5, and it was under the control of the beta-globin promoter on an NL43 base vector. The vector design itself is depicted here in reverse orientation. The treatment was given to 6 patients, aged 7 to 25, all with severe sickle cell anemia over the course of 2 years. Lab results showed that net hemoglobin F fraction increased from 5 to 12.7 picograms per cell, and it was further shown by hemoglobin F induction increasing to around 30% from around 10%. Hemolysis lessened and hemoglobin count increased net. Clinical results showed no acute negative reactions to treatment besides a single patient suffering from priapism who had already had priapism, and transfusions given to sustain quality of life for patients decreased in every single patient. Severe sickle cell related clinical events went to 0 in 5 of the 6 patients, with patient 6 also seeing a decrease.
So overall, the researchers found that the silencing of globin gene expression in adult human erythroid cells is dependent on the presence of the BCL11A gene. Therefore, this gene does influence fetal hemoglobin expression, where higher levels of fetal hemoglobin is associated with reduced BCL11A expression. Additionally, the opportunity for effective therapeutic interventions at different points in the pathogenic pathway strongly suggests that the combination of 2 or more drugs, each with a different mechanism of action, would be additive or perhaps even synergistic. The implication also works for other diseases that could benefit from gene silencing, such as the downregulation of a gene rather than the addition of a gene. And that's our presentation. Thank you very much for listening.